For our microscopy 2, entitled Structure of Plant Cell, before we begin, let me introduce our members. Bisak Tricia P, Cantillo Carlin C, Jayag John Peter C, Marvida Marian C, Payong Ellen Maya T, and Reyes Maria Kisha DB.
Objectives in this activity are to identify the basic parts of a plant cell and compare the shape and structure. Introduction. In this laboratory activity, we are going to identify the basic parts of the specimens, namely onion roots or allium cepa, hydrilla or elodea also known as digman, tomato or Solanum lycopersicum, and squash stem or trichomes, Cucurbita maxima. This activity aims to help us understand the different plant tissues and their appearance under the microscope.
For this activity, we used several plant specimens, which is onion roots, tomato skin, squash stem, and digman. For the tools, we prepared glass slides, cover slips, a dissecting kit, a microscope, medicine droppers, and staining solution iodine and carmine aceto. For the methods, first, we prepared the needed materials and specimens. Second, we cut a small portion of the specimens to be observed under the microscope. Then, we placed the cut portion on a glass slide, added a drop of iodine solution, also used carmine aceto solution in onion, covered it with a cover slip and observed it under the microscope. And lastly, we identify the basic part of the specimens. Next slide, result and discussion.
As we can see in the photo, the tomato skin showed large clear plant cells with visible cell wall, cytoplasm, and vacuoles. At higher magnification, the cell boundaries become clearer, confirming the organized structure of tomato's epidermal tissues. As we compare both images, we can confirm that observed samples showed typical tomato epidermal cells, including polygonal shapes, clear cell walls, and organized structures. This match verifies that tomato skin displays key plant cell features even at lower magnificence.
The squash trichomes we viewed under the microscope look almost identical to the online image. Both show the same trichomes body and epidermal base cell, with only slight differences in clarity due to our microscope's lower magnification.
In our microscope slide of the onion root tip, we were able to see the main cell structure such as the cell wall, cytoplasm, and in some cells, the nucleus. The stages of mitosis were visible, but the details were quite faint because the image was less sharp and the staining was lighter. While the online image provides the detailed high-resolution view that helps confirm what we observe.
In our microscope view of Digman, we could clearly see the cell wall and the chloroplast, but the image wasn't very sharp. The chloroplast appeared as small green structures, but their shape and arrangement were not very detailed due to limited magnification. In contrast, the online reference image showed the cell much more clearly. The chloroplast well-defined, clearly evenly distributed, and their arrangement inside the cell is easy to observe. The cell wall are also sharper and more distinct compared to our actual slide.
In conclusion, In this laboratory, we observe the structure of plant cells and the process of mitosis. We use specimens like onion, tomato, hydrilla, and squash stem to study different cells part under the microscope. Each specimens show unique shapes and features. We also examine onion roots, tips to identify the different stages of mitosis. So overall, the experiment helped us understand how plant cells function and how mitosis support plant growth and development.